Hi guys, good morning. Well, it's morning for me. I don't know what time you're watching this, but all right. We are going to go over the chapter 8 for cells today. I'm going to do lesson 8.1, which is called Life is Cellular, as well as lesson 8.2, which is the cell structure in this video. And then I'll post a second video for the other half. All right, so as we watched in the video the other day, Anton van Leeuwenhoek was the first to observe living microorganisms. All right, and what did he see? He saw those little tiny cells. All right, so the cell theory, we know there are 3 parts. So at this point, hopefully these are committed to memory and you can pick out the 3 main parts of the cell theory. The first one is all living things are made up of cells. The second one, and I think kind of the hardest one to remember, is that cells are the basic units of structure and function in living things. And then third, new cells are produced from existing cells through mitosis. Yeah. All right, so exploring the cell. Most microscopes use lenses to magnify the image of an object by focusing light or electrons. And we watched a interactivity video yesterday in the online book about the 3 different types of microscopes. So we're going to go through those really quick. Okay, there are 2 types of electron microscopes. We have TEM, transmission electron microscope, and scanning, SEM. All right, so if you look at these 3 images, one is a compound light microscope, which is the one we typically use in a biology class like yours. And let me move this down a little bit. Okay, so this would be an image through a light microscope. And these are all yeast cells, by the way. This is a transmission electron microscope and what the image would look like. And then scanning electron microscope. Okay, so scanning is going to cover the outside. You'll see like the surface area and any like special features. For example, I think in the video we watched, it was a fly's compound eye. And you can see the surface of everything really well. Whereas transmission, you can see through it. It bounces the electron beams through so that you can see the smaller organelles and the features inside of cells. All right, so cell size. Certain units of measurement are used for tiny objects such as cells. Okay, like we use micrometers. I think this arrow is supposed to be over here. Okay, so that unit, it looks like a fancy U and an M, is micrometers, and that's what we use for electron microscopes. So things that are super small are measured in micrometers, and then nanometers would be smaller. Okay. All right, so a chicken egg, we usually use like centimeters to measure that. A typical eukaryotic cell would be in that range there, 100 micrometers. Prokaryotic cells get even smaller, so like 1 micrometer. Mitochondrion, okay. And this is an amoeba called chaos. That's typically about 1 millimeter. We can use a light microscope, and we can kind of see those floating through the water. I've seen 1 amoeba in my life under a compound light microscope. And then a cold virus, like COVID, is only 1 nanometer. So those are so tiny. That's why we wear masks and why we want double layered masks and masks with filters in them, because they can travel through fabric because their particles are so small. So more layers, the better. And then, of course, DNA is even smaller than a virus. All right. So prokaryotes versus eukaryotes, we'll be talking about that more in class today. But prokaryotes are smaller bacterial cells, whereas eukaryotes are either plant or animal, and we're working on our project for plant and animal cells today. Look at all the organelles inside there. All right. Okay, so they all have a cell membrane. And cell membranes are something that all of them have. However, a nucleus is not something that a prokaryote has. Nuclei would be in animal and plant cells. Oh man, I went through lesson 2 already. I think I'm going to keep going. Yeah. All right, so we're on cell structure now. No, that is lesson 2. I misnumbered that. I apologize. We're on lesson 2, cell structure. Okay, so exploring cell organelles. We're going to look at the differences between plant and animal. Okay, so we know the nucleus controls most of the cell processes and it contains DNA. So at this point we know there's a nuclear envelope, hopefully. You probably don't remember this word, but nucleolus is on the inside, and chromatin will condense into chromosomes later on when we talk about mitosis. And there are nuclear pores, so that things like RNA can travel in and out, and we'll talk more about that in the later chapters. All right. Okay, organelles that build proteins. So proteins are assembled on ribosomes. Some proteins complete their assembly on the rough endoplasmic reticulum, and then the proteins will often be carried to the Golgi apparatus in something called a vesicle inside of a cell. So the nucleus doesn't just control the cell, it controls all the proteins that are made, and in turn, all those proteins are the traits that make you look the way you do and make your cells function properly. So it's not just that simple thing, it also helps control proteins. Okay, so organelles that build proteins continued. So the Golgi apparatus, we know then sorts and packages things. I like to think of this one as like a post office. Some people maybe put it as a factory. Vesicles are shipped to their final destination. So any other cell or any part of the body that might need proteins or lipids. Okay, so review. Starts out in the nucleus. All right, that's where they're made. The rough endoplasmic reticulum is where they can go. R for ribosome rough. All right. Sometimes we get free ribosomes floating throughout the cytoplasm of the cell. They can travel through the smooth endoplasmic reticulum and leave it through vesicles. And then also the vesicles would then bring it to the Golgi apparatus, that it might go through some new chemical changes and then modifications, and then move on out to be a different protein that can be used for other cells. And that is literally all you need to know about proteins at this stage. We will talk more about protein synthesis when we get to the RNA chapter. Okay, so vacuoles, vesicles, and lysosomes. What are these? What do they do? So the central vacuole is right there, okay? It is what contains a lot of the water and the nutrients plants need. So you won't typically see large vacuoles in animal cells. They're contractile vacuoles, which are very, very small compared to what a plant would need. Okay, and if you use a microscope and a transmission microscope, you can see the inside of cells have little roadways. That's called the cytoskeleton. Though that is helps with support and structure, it helps things travel throughout the cells. And also microtubules, which are very common in plants, is another cytoskeleton or type of support structure in plant cells. Okay, so this is showing you a cross-section of a plant. Okay, and then chloroplasts. So we do need to know that chloroplasts capture energy from sunlight and convert it into food that contains chemical energy. Later on we'll learn this one even more in depth. They are found in plants, and some organisms such as algae do photosynthesize with chloroplasts as well. And they convert chemical energy. So they're using sunlight to do that. All right, so a very similar organelle for animal cells is mitochondria. They convert chemical energy from food into compounds the cell can use. We call this process cellular respiration, which will be in few chapters. Cellular power plants, the mitochondria convert chemical energy stored in food into a form that can be easily accessed by the cell in the form of ATP. Okay. And this is what the inside of the mitochondria looks like. It has a lot of surface area, just kind of like similar structure to what your brain looks like. So that there's lots of surface area and lots of places for ATP to be made. So every single cell in your body, it's supposed to be pointing right there. Okay, every single cell in your body has mitochondria in them for energy. Okay, and both of them do have cell membranes. All three of them have cell membranes, but prokaryotes and plant cells will have cell walls. Okay, cell membranes are very important structures. We do need to spend a little time learning about this. All right, so they do have what they call a phospholipid bilayer. Okay, and you can see they kind of look like, I don't know, your kindergarten drawings of people with a head and two legs sticking out the bottom. So that would be called a lipid. They have a hydrophilic head and a hydrophobic tail. Hydrophobic is on the inside because it repels water, whereas the hydrophilic head is okay with water hitting the surface, and other proteins and nutrients might hit the surface, but it's going to be a protective layer because of that hydrophobic tail and the hydrophilic head. So you can see there are membrane proteins and channel proteins that are in there to allow certain, to help with homeostasis, to allow certain nutrients in and out of every cell in your body, and you have trillions of cells in your body. All right, so here's the cell membrane out here. All right, but we are zoomed in on it looking at what it looks like. All right, it is very pliable and you know, a very movable substance, so that those cells can be compact and fit into shapes of organs and things like that. All right, so this would be the inside of the cell where the cytoplasm is, and that's the outside. Guess I should have been clicking. All right, and a lot of times there are carbohydrate chains directly embedded, as well as those membrane proteins. All right. This is 8.3. I'm going to stop here, and I will record the second half of the lecture later. Comment below if you have any questions, guys.